Hi everybody, welcome back. Today we're going to be discussing structures of the brain. And we're going to start with the meninges. So remember, we've already talked about the meninges in the spinal cord. And similar to the meninges in the spinal cord, the meninges of the brain also has 3 layers. You can see them in the spinal cord here. We have the Dura Mater, the Arachnoid Mater, and the Pia Mater. The function of the meninges are to protect and cover the brain, as well as to contain the cerebral spinal fluid. And remember, the cerebral spinal fluid in the spinal cord runs in the subarachnoid space, so the space underneath the arachnoid mater. Today we're going to look at what's going on in the brain. And not only do the meninges form coverings in the brain and have 3 layers, they are also responsible for forming partitions within parts of the brain. So for example, we've talked about the brain model, which I have here, and we know that this sort of area in between the 2 halves is the longitudinal fissure. One of the jobs of the meninges in the brain is to line those spaces in the brain. So this is a picture, this looks like a, I can't actually tell, this looks like a posterior view. And the very top where the longitudinal fissure is, you can see that the meninges actually dive down in between the 2 lobes of the brain. That particular division is called the Faux Cerebelli. We have another division right in here where the meninges sort of line the separation between the cerebrum and the cerebellum. That's called the tentorium cerebelli. And then the last one is between the 2 lobes, get that in there, between the 2 lobes of the cerebellum. So right in between those 2 lobes, sort of between where you can see the arborvitae in this particular picture, that is called the Faux Cerebelli. So it has 3 different areas where it's separating and dividing parts of the brain. Let's look a little bit closer at this Faux Cerebelli. So here we are looking at a close-up of the longitudinal fissure. And one other difference between the meninges of the spinal cord and the meninges in the brain is that there's no epidural space here. So remember, around the spinal cord, the dura formed a covering, and outside the covering and between the inside of the vertebral arch, there was some fat that we called the epidural space. Here, the dura lies right up against the inside of the skull. I'm going to point out the Dura Mater is right here. It's shown in white, and I'm going to sort of highlight it with my blue pen. And you can see that it's literally right up underneath the parietal bones. You can see the suture between the 2 parietal bones right here, the sagittal suture, and the skin lays on top of them. And the dura is right up next to it. Underneath the Dura Mater is the Arachnoid Mater, excuse me. And that's shown here in purple. I'm just going to sort of outline it. And you can see the Arachnoid Mater actually forms little, they look like little trees, but it's a way to send some of that cerebral spinal fluid that's in the subarachnoid space up into the Dural Sinus. So here is the Arachnoid Mater. And the Pia Mater, I'm kind of in trouble because I don't have pink, but we're going to do red. The Pia Mater is very closely associated with the gray matter of the skull, and you can see it on both sides here. You can see the gray matter of the cerebrum is on the outside, and the white matter of the myelinated tracts is on the inside. In between the Arachnoid Mater and the Pia Mater are little sort of, they almost look like spider webs, which kind of makes sense since it's called the Arachnoid Mater. And that is where the cerebral spinal fluid is going to circulate around in the brain. Now, if you look closely, the Dura Mater actually splits into two layers. So we have this layer that's out here. We actually call this the periosteal layer. And it makes sense. It's called periosteal because it's sort of right around or right below the skull. This lower layer runs alongside, right on top of the Arachnoid Mater, and that layer is called the Meningeal Layer. And that also makes sense because it's running right along the top of the rest of the meninges. And then it dives deep, deep, deep into the longitudinal fissure to make that faux cerebri. Now, the space between the periosteal layer and the meningeal layer forms an open space, a sinus. And this is actually called the Dural Sinus. And it's filled with venous blood. So this is blood that is heading back to the heart. So it's already dropped off all its nutrients and oxygen. It picked up waste products and CO2, and it's going back to the heart to be pumped back to the lungs and then back out to the body again. And so what happens here is these arachnoid granulations, these little areas where cerebral spinal fluid can come through and poke up and circulate around in these spaces, those are called arachnoid granulations. And those contain cerebral spinal fluid. And so nutrients and things that are being carried in the cerebral spinal fluid can actually leave the cerebral spinal fluid and go into the venous blood to be carried away from the brain and back to the heart. So this is part of the circulatory system of the brain. We can control very well what's coming into and what's forming the cerebral spinal fluid. And so that helps as a layer of protection to keep things out of the brain when we don't want them. So let's talk a little bit more about where and how cerebral spinal fluid is made. So remember in one of the videos recently, we talked about the structures of the brain, and I showed you this funny looking structure. This is a negative space mold. So basically, these are the lateral ventricles, third ventricle, and fourth ventricle of the brain. These are spaces within the brain where fluid circulates. And if you look at this piece of paper, you can see where they are. So here and here are the lateral ventricles. Draining those lateral ventricles is a little tube called the Interventricular Foramen. So that's written right here. And you can see that on here. So this space and this space connect the lateral ventricles with the third ventricle, which is right about there. The third ventricle is going to drain into the fourth ventricle by way of a little tube called the cerebral aqueduct. So all of four of the ventricles are connected, and cerebral spinal fluid flows through them. All four of the ventricles have a specialized structure called the choroid plexus. The choroid plexus is made out of a kind of neuroglial cell called the Ependymal cell. So let's do a quick review of the four types of neuroglia in the central nervous system. All right, so the first ones are the oligodendrocytes. These are cells that form the myelin sheath in the CNS. Our next type of cell is an astrocyte. Astrocytes are kind of the janitor of the cell. They help to support and clean up after the neurons, and they also help to form the blood-brain barrier, which we're going to talk about in a few minutes. The next type of cell are the microglia. These are cells that help to form the immune system that is specific for the central nervous system. And the last type of cell are the ependymal cells. The ependymal cells line the ventricles and form cerebrospinal fluid. So I have a picture here of a blood capillary that is coming in and bringing blood to the brain. And the Pia Mater, the inner meninges, is going to line this. So you can see the simple squamous epithelial cells of the Pia Mater. Lining the ventricles, you'll see these ependymal cells. The ependymal cells have special tight junctions. And remember, a tight junction is a type of intercellular junction where the cells are literally fused together. So they're waterproof. So the material in the blood is not going to be able to get through the space between the cells. We prevent that. Everything that comes from the blood and becomes CSF gets filtered. It has to go through the plasma membrane of these ependymal cells. So as we filter this liquid, it's going to be mostly water that's coming through. This filtration of substances from the blood to the cerebrospinal fluid prevents potentially harmful pathogens and substances coming into the brain and circulating through that ventricular system. So remember, I told you the ependymal cells are the cells that are going to filter the blood and line the ventricles. There are specialized areas called the choroid plexus that exist in each of the ventricles. So there's one in each of the two lateral ventricles, there's one in the third ventricle, and there is one in the fourth ventricle as well. There's ependymal cells that line this entire area, and some of those ependymal cells form something called a choroid plexus that filters the blood and allows some metabolites, some nutrients, and mostly water to enter the ventricles. Now, there is a specific set of places where the cerebral spinal fluid is made and flows. So the lateral ventricles is where the cerebral spinal fluid is first created. It flows through the interventricular foramen, which you can see right here. So it's made in the lateral ventricles, flows through that interventricular foramen, enters the third ventricle, which is right about here. More cerebral spinal fluid is made in the third ventricle by the choroid plexus there. The fluid will flow down through the cerebral aqueduct into the fourth ventricle, which is right here. So here's lateral ventricle, here's third ventricle, here's the fourth ventricle. There is another choroid plexus in the fourth ventricle where yet more cerebral spinal fluid is being made by filtering blood. And then the cerebral spinal fluid has a couple of places it can go. It can enter the median aperture, and it'll flow this way around the cerebellum, or it can go through a lateral, one of two lateral apertures, in which case it will be flowing down through the spinal cord. And remember, the cerebral spinal fluid is going to flow in that subarachnoid space in both the spinal cord and the brain. As it flows and comes back up from the spinal cord, you can see that it's entering that subarachnoid space. It comes along and it fills in those arachnoid granulations or the arachnoid villus. And waste products and metabolites will leave the cerebral spinal fluid and go back into that dural sinus. The blood will take those waste products back to the heart to reoxygenate the blood and send it circulating around the body again. So the point of this is that we make cerebral spinal fluid in all 4 of the ventricles. It circulates in the subarachnoid space of the brain and the spinal cord. It eventually leaves that subarachnoid space by diffusing across either the arachnoid villi or the arachnoid granulations. You'll see both of those in your textbook. And then it will enter the venous blood and head back out to the heart. Because the choroid plexus are constantly filtering blood and the CSF is constantly being removed through the arachnoid villi, the volume of cerebrospinal fluid stays constant at about 80 to 150 milliliters. So even though we make about 500 milliliters per day of cerebrospinal fluid, the volume at any one time stays constant. If you were to get a blockage somewhere in the brain so that this flow was impeded, it would cause a backup of cerebrospinal fluid, and that's called hydrocephalus, also known as water on the brain. But really it's not water, it's just a backup of cerebral spinal fluid. What this causes is increased pressure on the cranium and the spinal cord. Now that's the cerebral spinal fluid, and we have a special barrier called the Csf Blood Barrier. We also have something called the blood-brain barrier. And instead of ependymal cells and tight junctions in the ependymal cells, in this case, we're talking about astrocytes. So astrocytes are those glial cells that help to support and clean up, and they form the blood-brain barrier. They have these projections that wrap around the blood capillaries that are in the brain. Those blood capillaries are made out of simple squamous epithelium, just like all capillaries are made out of simple squamous epithelium. However, these ones have, you guessed it, tight junctions. And remember, tight junctions is the type of intercellular junctions that form a watertight barrier. So as blood comes through the blood capillaries, certain things can leave because they're able to diffuse across the plasma membrane of the simple squamous epithelium. The things that can leave the blood and enter the brain are things like oxygen. CO2 can go the other way. Alcohol, barbiturates, nicotine, caffeine. All of these things are able to diffuse right through the plasma membrane. Remember, when we talk about transport across the membrane, these were all the things that are lipid soluble, nonpolar, and small. Other things like glucose cannot diffuse directly across the membrane, but they can be carried across via facilitated diffusion. And it's pretty good that we can do that because the brain can only use glucose to make energy. It cannot use fat or anything else. So glucose is really important for the brain. An interruption of blood flow to the brain reduces the amount of glucose available for the neurons to make ATP. When blood flow to the brain slows or is stopped, or there's a lack of glucose in the blood, dizziness or a lack of consciousness can occur. A lack of glucose or oxygen to the brain can very quickly, within about 4 minutes, lead to permanent damage. So the astrocytes wrap themselves around the capillaries. The capillaries themselves have tight junctions that limit the materials that can come in and out of the brain. So while things that get you drunk or make you high or make you feel the effect of stimulants can pass, many things such as antibiotics and various proteins cannot pass the blood-brain barrier, and so our brain is protected from those substances that might be traveling in the blood. That's it for today. See you in class.